Hi everyone, today we'll be covering the IGCSE Human Biology past paper. This is from 2023 January and this is paper 1. So you're given an hour and 40, okay. You guys are given an hour and 45 minutes to do this, and the total marks for this paper is 90, and this basically tests on the entire the entire syllabus. All right, let's start off with question 1. Okay, question 1. The diagram shows a human eye. So before we start matching up the structure to its function, we'll just go over the structures here. So over here, Z is the retina. So the retina consists of light receptors that can detect light. Over here we have the lens. So this helps refract light. So if light is coming in here, the lens refracts the light so that it lands on the retina. And Y here is your iris. So let's go on to this part here. Draw one line from each structure to its correct function. So let's start off with structure X. Structure X is the lens, and the lens is responsible for refracting light. Structure Y here is the iris, and the iris is responsible for controlling the size of your pupils. So your pupils can dilate, which means they get larger, or your pupils can get smaller, in which case they would constrict. And so the purpose of your iris is to ensure that not too much light comes in, because you don't want your eyes to get damaged. All right, and structure Z over here, that is the retina, and the retina here detects the light rays. Okay, so that's the answer for question A1. Let's look at A2. Most humans have stereoscopic vision. Which of these statements occur because of stereoscopic vision? So stereoscopic would mean that you have good depth perception. So this is A. And part B. So the diagram shows a healthy eye and an eye with cataract. Describe the cause of cataracts. Okay, so this is caused by the lens being cloudy. So this is caused by the lens becoming cloudy. All right, state one treatment for cataracts. So there's not much. There's only surgery to replace the lens with perhaps an artificial lens. So you can say surgery to replace the lens. Okay. Part 3. It is estimated that 39 million people in the world are blind. Of these people, 43 people are blind because of cataracts. Determine the estimated number of people who are blind because of cataracts. So 39 million people, that would be this many zeros, and then we're just going to times that by 0.43, which is the same as 43%. So let's just use the calculator. Times 0.43, and we have 16 million people. So that's 16770 and then 000. 770000. Some people are blind because of a vitamin deficiency. Name the vitamin that is needed to prevent this type of blindness, and that would be vitamin A. Vitamin A. Okay, let's move on to question 2. So part A, a human sperm cell has a tail, a midpiece containing organelles, and a head with a nucleus. Give the function of the tail. So the tail enables the sperm to swim. So it enables the sperm, to swim. Okay. Which of these found in the midpiece releases energy? That would be the mitochondria. And the process of meiosis is involved in the production of sperm. Explain the importance of meiosis in sperm production. Okay, so I've included a diagram here, but let me just make this one a little bit bigger. So on the right, we have meiosis. And so meiosis ensures that you form these haploid cells, which would contain half of the number of chromosomes that we have in our body cell. So for example, in our body cell, we have 46 chromosomes. And at the end of meiosis, you would have 4 haploid daughter cells. So each of them would have 23 chromosomes instead of 46. So that if you have, let's say, a sperm and an egg, each of them both have 23 chromosomes, they would undergo fertilization together to form a zygote, which would have 46 chromosomes. So this establishes the, this establishes back the number of chromosomes that we're supposed to have. Okay, so I'm just going to erase this. And how do I make this one smaller? Okay, let's just make this one smaller. Oh, we've got some numbers to remove. So, meiosis halves the number of chromosomes from diploid to haploid. This ensures that the diploid number is restored. When fertilization occurs. Another thing that meiosis also does is that it shuffles around the alleles, so that introduces genetic variation. So in meiosis, you would have, let's say this is one chromosome, this is another, so these are the homologous chromosome, one from mom, one from dad. And what they would do is they would swap out. So let's say this is the maternal chromosome and this is the paternal chromosome. So what would happen is they would swap out parts of it during what we call crossing over. So I'm just going to attempt to draw. Maybe I should have included a diagram. Okay, so you can see that they've swapped this part out. Right, and that's what happens in meiosis. So this ensures that, you know, our siblings don't have the same genetic material that we do. Okay, so meiosis also introduces genetic variation. Okay, let's go on to the next page. Complete the genetic diagram to show how the egg of a person is determined by the chromosomes in the nucleus of the egg and the nucleus of the sperm. Okay, so for eggs, so eggs would come from the female, and females would only have XX chromosomes, whereas sperms come from males, so then 50% of the sperm would have an X chromosome, and 50% of the sperm would have a Y chromosome. So this is how we would fill up this diagram. Okay, so you can see here, right, there's a 50% chance of the offspring being male and 50% chance of the offspring being female. All right, the last part. The sperm head has to enter the egg cell so that the nuclei can fuse. Describe how the sperm head enters the egg cell. So at the tip of the sperm, where the head is, you have this thing called the acrosome. So the acrosome contains digestive enzymes that would break down the egg cell membrane. So actually, let me just include a diagram so that you can see. Okay, so you can see here at the head of the chromosome, we have this thing called the acrosome, and the acrosome is here. Okay, let me just put this to the side. So describe how the sperm head enters the egg cell. So the sperm head contains the acrosome, which contains digestive enzymes that would break down the egg cell membrane. Okay, let's go on to question 3. The diagram represents the effect of bile on a large fat droplet. Where is bile produced? So bile here is produced in the liver. So it's produced in the liver and then stored in the gallbladder. Okay, describe the role of bile in the digestion of fat. So we can see here this is bile, and here we have a large fat droplet. So this ensures that the bile is responsible for ensuring that the fat droplets are smaller. So you can see they're much smaller. And the reason why that's a good thing is that it increases the surface area for enzymes like lipase, which are enzymes that break down fat. So it increases the surface area for lipase to work on them. So that's one of the functions of bile. The other function is to neutralize the pH. So if you recall, the stomach is very acidic, and then when the digested material from the stomach enters the small intestines, the bile is used to neutralize the pH, so it's no longer acidic. So those are the two functions. So number one is that it emulsifies fat. So this is what emulsifying does. So bile is responsible for emulsifying fats, which increases the surface area for lipase to break down or digest. It is also responsible for neutralizing the digested material coming from the stomach. Okay, so let's look at the mark breakdown. So 1 mark for emulsifying, 1 mark for increase the surface area, another mark for lipases to break down the fats, and another mark for neutralizing the digested material, neutralizing the pH. Okay, so that's 4 marks. Let's move on to part B. Alright, 3 food tests are used on a solution of food. The table shows the reagent used and the color of the food solution after each test. So let's go through them one by one. So iodine solution is responsible for testing for the presence of starch. The original color, I'm just going to write it down here, is orange-brown, and it is blue-black when starch is present. The second one is the Biret solution, so that's responsible for testing for proteins. So the original color is blue, and when proteins are present, it would turn purple or lilac. And then Benedict's solution tests for reducing sugar, so the initial color is actually blue. And if reducing sugars are present, then it will turn red. If there is a little bit of reducing sugars, then it will turn orange or yellow, green, orange, or yellow. But if there's a lot, then it would turn red. Yeah, so one little trick that I use to remember them is that both Biret and Benedict start with the letter B, and they both start off as being blue. Okay, so part 1, describe what the food tests show about the dietary components of the food. Well, it shows that number 1, it doesn't contain starch. It shows that it does contain starch because it's blue-black, right? That it does contain starch and protein because of the Biret solution, but does not contain reducing sugar. Reducing sugar. Okay. The next part, saliva is added to a new sample of food solution and the mixture is kept at 37 degrees for 10 minutes. The food tests are then repeated and the solution turns brick red when tested with Benedict's reagent. Explain the effect of saliva on the food solution. So before it was blue and then it turned brick red. So again, brick red is when you have the presence of a reducing sugar. Usually it would be, at least for your syllabus, it'll be glucose, right? Or something like maltose, for example. So your saliva actually contains this enzyme called amylase, and amylase is responsible, let me just write this down. So if you have starch in the food that we eat, amylase is the enzyme that is responsible for breaking it down to maltose. All right, so we can write that down here. The saliva contains amylase, which breaks down the starch into maltose. Okay, so the 3 marks would be, number 1 is amylase, another one is breaks down the starch, and then the 3rd mark is into maltose, or you could say glucose as well. Okay, now let's look at part C. Food is digested as it moves along the alimentary canal. So the alimentary canal is just basically the path which your food takes from your mouth all the way to your anus. This process is helped by a diet containing fiber. Explain how food is moved along the alimentary canal and how fiber helps this process. Okay, so let's make this image larger. So this is basically peristalsis. So here we have a diagram of peristalsis. We have these circular muscles that are contracting and they're pushing the bolus down the alimentary canal. So it's similar to us squeezing toothpaste out of the little container. So it would happen, and how does fiber help with this process? Well, fiber kind of acts like a sponge. It kind of absorbs water, and that adds bulk so that it makes it easier for the muscles to kind of contract. So it helps stimulates this process of peristalsis. So let me just make this one smaller. Let me just put this one down here. Okay, so the food bolus moves along the via peristalsis. This is when the circular muscles contract to push the bolus. So then we have to talk about how the fiber helps, right? So the fiber. Another thing to note is that the fiber does not get broken down, so it's able to absorb all this water. So the fiber does not get broken down. Okay. It absorbs water, which has bulk and therefore stimulates the muscles to contract. Let's go over the marks here. So peristalsis is one mark, of the food bolus is another. We have your muscles contract to push the bolus, that's a third mark. Does not get broken down is another mark. Adds bulk is another mark. And stimulate the muscles to contract will be the last mark. Okay, let's move on to the next question. Question 4. Typhoid is caused by Salmonella typhi bacteria. These bacteria are spread by houseflies. The photographs show Salmonella typhi magnified 30,000 times. Okay, so part 1, it says each bacteria is surrounded by cell structure Y. That would be the cell wall. So all bacteria have a cell wall. The next part here on the photograph, bacterium X has a length of 55 millimeters. Calculate the actual length of the bacteria in micrometers. Okay, so the equation for this is magnification equals image size over actual size. We're given the magnification up here, so that is 30,000. So let's rearrange this equation so that A is on one side. So that would be A. This would be I over M. So image over magnification. So the image size is 55 millimeters. And then we know the magnification is 30,000. So let's calculate that first. So 55 divided by 30,000, that would be, okay, 0.001833. Now, this is currently in millimeters, but it's actually asking you to specify it in micrometers. And it says here that 1 millimeter equals 1,000 micrometers. So we just need to times this by 1,000. So let's do that. Let's times that by 1,000, and we get 1.83. 1.83 micrometers. So 1.83. Okay, let's move on to part B. A team of scientists found that the legs of houseflies have hundreds of different species of bacteria on them. Oh God. Part 1, flies in towns and cities carry more bacteria than flies that are found in the countryside. Suggest a reason for this observation. Well, because towns and cities are a lot more populated, they might tend to have a lot more sewage or tend to have a lot more rubbish, for example. So we can say this is because they are more crowded and would tend to have more sewage. So this is actually 2 points here. So one of them is crowded, the other one is sewage, but you only get 1 mark for this question. All right, part 2. One species of bacteria identified as Helicobacter pylori, which causes stomach ulcers. Explain how houseflies could increase the transmission of Helicobacter pylori. Okay, so because it is a disease that causes stomach ulcers, it is possible that when a person poops it out or feces or has feces, that these feces would contain the Helicobacter pylori bacteria. And you could have a housefly that could land on the feces and then could possibly land on someone's food. And when someone ingests that food, then the Helicobacter pylori would be transferred from the food to another person's stomach. Okay, so explain how houseflies could increase the transmission of Helicobacter pylori. So we could say the houseflies may land on feces containing Helicobacter pylori and could land on a person's food. This person could ingest the contaminated food. So 1 mark for land on feces, another mark for transferring it onto food, and then the 3rd mark would be ingesting the contaminated food. Okay, let's move on to part C. The diagram shows the levels of typhoid across the world. Give reasons for the global distribution of typhoid. So there are 2 ways in which typhoid can spread throughout the human population. One of them is through houseflies, and the other one is quite similar to cholera. So you would have a person that would be infected with typhoid and their feces would contain the salmonella bacteria, and that could get into the water. And then if you don't have proper sewage treatment, people might drink the contaminated water or cook with the contaminated water. So some of the reasons that you can say here is that, okay, so give reasons for the global distribution of typhoid. So 1 reason is due to a lack of sewage treatment. Another one that you can also say is lack of healthcare as well. Maybe due to lack of healthcare. Other things that you can also say is that it looks like the distribution is more or less in more developing countries, and these places are also quite highly populated as well. Okay, let's move on to part 2. Some strains of bacteria that cause typhoid are resistant to antibiotics. Discuss how resistance to antibiotics develops and the effect this has on the treatment of typhoid. So let me just zoom into this picture here. Okay, so this question asks about antibiotic resistance. So for example, you might have a population of bacteria. Let's say most of them are normal and maybe you have 1 or 2 or several bacteria that are resistant to the antibiotics. So if you expose them, so this is here, so if you expose them to antibiotics, what would happen is that the ones that are not resistant, they would die. And the ones that are resistant, they would survive. And what happens now is that, hey, there's a lot more space for them to thrive. There's a lot more resources for them to live on now that most of the population is dead. So then they're able to thrive. They're able to grow in population. Another thing about antibiotic resistance is that with bacteria, they can transfer their genetic material or share their antibiotic resistant genes through plasmids. We can see that there as well. So let me just make this one smaller again. Put this here. Okay. Resistant. Oh, I need to mention that antibiotic resistant bacteria can arise due to mutation. So let me just write that down. So antibiotic resistant bacteria can arise due to mutation. When exposed to the antibiotic, they are able to survive, reproduce, and pass on their alleles to their offspring. Offspring, making the offspring resistant to the antibiotic. Okay, and it also asks for how this would have an effect on the treatment of typhoid. Well, if you have a lot of these bacteria that are more resistant, you would end up having more serious infections. This could lead to more serious cases of infection. Let me see if that's enough marks. So one of them, so one mark for due to mutation. So antibiotic resistance is due to mutation. They are able to survive. That's another mark. They're able to reproduce. That's a third mark. And their offspring is resistant to the antibiotic. That is a fourth mark. More serious cases of infection will be a fifth mark. Other things you can also say is a lot of the times this could happen because the patient does not finish their antibiotic course. So for example, let's say the doctor says, oh, you need to take like 2 pills a day for, let's say, 10 days. but the patient doesn't do it for the whole 10 days. That could lead to the development of these antibiotic resistance bacteria. Okay, so we'll cover question 5 in the next video.